Okay, good morning, students. Let us continue the yesterday's class, that is protein chemistry. So, in the previous class, already we have discussed about the how like what are the elements which are made up of proteins like carbon, hydrogen, oxygen, nitrogen, and functions we have discussed. And you all know that proteins are like polymers of amino acids. Like proteins are linear sequence of amino acids, and amino acids have been joined by peptide bonds. Now there are 20 amino acids are which maybe out of 300, 20 may participate in protein synthesis. Those we can call it as standard amino acids or primary amino acids, and those 20 amino acids has been classified into, and you know the common structural feature of an amino acid. One side there will be to the alpha carbon, there will be amino group, carboxyl group, and third valency of the alpha carbon has been attached to hydrogen, and the difference between among all the amino acids occur at this R represents a side chain. Now here this is the general structure, and amino acids are these are the names of the 20 standard amino acids. They have been designated, assigned as 3-letter abbreviation. Now here these 20 amino acids are further classified into four classes based on the structure of the side chain, based on the solubility, polarity, based on the nutritional requirement, and based on the metabolic fate. And already you know that based on the charge, based on the side chain structure, amino acids have been classified into neutral amino acids, acidic and basic, and most of the amino acids are neutral amino acids because as they are monoamino, monocarboxylic acid, and these neutral amino acids based again further subdivided into aliphatic, hydroxy, sulfur containing, aromatic and amino acids. Already this we have discussed. Acidic amino acids mean they may contain additional carboxyl group in the side chain, aspartic acid and glutamic acid. Basic amino acids, they may contain additional amino groups in their side chain. Examples of basic amino acids are lysine, arginine and histidine. Now based on the solubility, second classification is based on the solubility. These amino acids are of two types like polar and non-polar. Polar amino acids are hydrophilic, and here they are readily interact with the water, and non-polar amino acids are poor interaction with the water since they are hydrophobic, and non-polar amino acids are alanine, valine and leucine. In these polar amino acids, they may in the side chain they have without charge on R group are there, and few polar amino acids they may carry charge. So charge may be positive charge and negative charge. Positively charged polar amino acids are basic amino acids, and negatively charged polar amino acids are acidic amino acids, and polar amino acids which has no charge are simplest amino acid glycine, serine and threonine. Now coming to the third one, classification based on the nutritional requirement. Nutritional requirement is here means, so based on the see here, based on the nutritional intake of that particular amino acid, these 20 standard amino acids has been classified into two types, essential and non-essential amino acid. Based on the nutritional requirement, amino acids are of two types, essential or indispensable amino acids, or else second type is non-essential or dispensable amino acids. Essential amino acids means here you know that these amino acids cannot be synthesized by human beings. So the entire amount is required by the body has to be obtained through diet. So essential amino acids, why they are called essential? As the name only indicates, they cannot be synthesized by human beings. So they have to be taken in food for normal growth. Out of the 20 amino acids, 10 are essential amino acids. Examples of the 10 means the amino acids which are essential are isoleucine, leucine, threonine, lysine, methionine, phenylalanine, tryptophan and valine. Okay, and here even histidine and arginine also generally we do see here, histidine and arginine also generally we will include under essential amino acids only. So out of 20, 10 are essential. But here these, especially the last two, histidine and arginine, are semi-essential or partially essential. Why they are semi-essential means, as in the growing stage, growing children, they require them in the food, but they are not essential for the adults. So that's what histidine and arginine are called semi-essential. Essential or 10 only, including this histidine and arginine, but especially histidine and arginine are semi-essential because growing children, they need these two amino acids in the food, but their histidine and arginine is non-essential in the adult individual. So that's what these two are called semi-essential amino acid. Next one, that why the adults, why it is not required in the adults means that some amount which is synthesized in the body is sufficient to meet the requirement of the adults. So that's what in the adults histidine and arginine is not required to take in the diet. But children, as they're in the growing stage, they need more of this amino acids. So histidine and arginine has to be included in the diet in the children's diet, and the other 10 amino acids are non-essential. So these carbon skeletons of these amino acids can be synthesized in our body from various sources. So these amino acids are called non-essential amino acid. If you know the names of the 20 amino acid, okay, first you find out the names of essential amino acid. The remaining 10 amino acids are non-essential. So that is the third classification based on the nutrition: essential or indispensable, non-essential or dispensable amino acid. Under essential only, one semi-essential are two are semi-essential: histidine and arginine. Then coming to the next one is fourth classification, that is based on the metabolic fate. So based on the metabolic fate means amino proteins, once it is being digested in the body, it may be proteins are digested or hydrolyzed to amino acids. Once amino acids enter into your blood circulation, they may undergo further catabolism. Catabolism means nothing but degradation. So in the course of their degradation, the amino acids may be, few amino acids may be converted to ketone bodies, or few amino acids may be converted to glucose, or few amino acids may be converted to both glucose and ketone bodies. So like that, based on the metabolic fate. Metabolic fate means amino acids when they are catabolized in the blood. Okay, when they are catabolized means when they are degraded, they may be converted to ketone bodies or glucose, or else both glucose and ketone body. So based on their metabolic fate, how they will be converted, the carbon skeleton during the degradation of the amino acids, how they will be converted. So based on their metabolic fate, amino acids are classified into three types: ketogenic, glucogenic, and both glucogenic and ketogenic. Okay, based on the metabolic fate, amino acids are of three types: purely ketogenic, purely glucogenic, and both glucogenic and ketogenic. Purely ketogenic means from that particular amino acid, if it is degraded, finally it will be converted to ketone bodies. So those amino acids you can call it as ketogenic. Out of the 20, only one amino acid is ketogenic, that is leucine. And the next one is ketogenic and glucogenic means, first let me finish up this. The second type is glucogenic. Glucogenic means the carbon skeleton of the amino acids during their degradation, if they may convert into glucose, those amino acids are called glucogenic. Please note down, out of the 20, most of the amino acids, 14 amino acids are glucogenic. I think you are getting. So excess intake of the protein also increases the carbohydrate glucose concentration. How means? Because most of the amino acids, 14 amino acids, are glucogenic. Okay, excess protein intake is there means more availability of the amino acids, and we know that most of the amino acids are glucogenic. So in the course of their degradation, they may be converted to glucose only. And the next one is See here, the both glucogenic and ketogenic means the carbon skeleton of these amino acids may serve as the precursor for the synthesis of both glucose and ketone bodies. Those are called both glucogenic and ketogenic. Out of the 25 amino acids are both glucogenic and ketogenic. Examples are aromatic amino acids. All three aromatic amino acids, phenylalanine, tyrosine, tryptophan, isoleucine and lysine. All these are metabolic weight of these 5 amino acids are both glucogenic and ketogenic. Only one is ketogenic, that is leucine, and the rest of the 14 amino acids are glucogenic. Okay, like this amino acids has been classified into various different types based on their metabolic weight, glucogenic, ketogenic, and both glucogenic and ketogenic, and based on their nutrition, essential, semi-essential and non-essential, and based on the structure, you know, neutral, acidic and basic, and based on their polarity, polar groups, polar amino acids, hydrophobic and non-polar hydrophilic amino acids, etc. So like polar and non-polar amino acids. So that's all about the classification of the 20 standard amino acids. Now coming to the next one, non-standard amino acids or modified amino acids. Non-standard or modified amino acids means here these are the amino acids which are formed by the modification of the one of the 20 amino acids. Okay, so these are the standard amino acids are 20 only, but what are these non-standard amino acids means the one of the 20 standard amino acids they may undergo further modifications after incorporating into the proteins. So those you can call it as modified amino acids. Okay, standard amino acids only, after incorporating into the protein they may be modified. Modified means modified by the addition of the extra additional groups. So those amino acids are non-standard amino acids or modified amino acids. So examples of this modified amino acids are, please note down, this is an important short answer question. Hydroxyproline, that means here the proline is the addition of hydroxyl group to the proline to form hydroxyproline, that is the modification which has taken place here. And hydroxylysine, these two are the modified proteins formed by the hydroxylation of the proline and lysine to form hydroxyproline and hydroxylysine. And these hydroxyproline and hydroxylysine, they are the structural components of the connective tissue, that protein, that is collagen. Okay, see here this hydroxyproline and hydroxylysine, as they are modified proteins, but they will perform various different functions. Okay, these modified amino acids, they may be perform several different functions. So hydroxyproline and hydroxylysine are the constituents of the connective tissue protein, that is collagen. And the next one is gamma carboxyglutamate, and this gamma carboxyglutamate is formed by the gamma carboxylation of the glutamate. Especially this is quite common in the side chains of the blood clotting factors, especially prothrombin. Okay, gamma carboxyglutamate means here how it is being formed by the gamma carboxylation of the glutamate. This is common common in the proteins of the blood clotting factors. Okay, next one, so blood clotting factors which involved in the clotting of the blood. And next one, cysteine. Cysteine is the modified amino acid which is formed by the oxidation of the two cysteine amino acids. Okay, now here cysteine is the amino acid. So you know that cysteine is a sulfur containing amino acid. If you see the structure of the cysteine, sulfohydral groups are there. So like that, oxidation of the sulfohydral groups of the two cysteine molecules to form cysteine. Okay, cysteine, two molecules of cysteine on oxidation to form cysteine. So that means sulfohydral groups of these two cysteine amino acids linked to each other to form the dimer disulfide to form the cysteine. And here the cysteine is the which gives stability to the three-dimensional structure. Okay, the cysteine is formed by the condensation, oxidation of the two amino acids cysteine. So there is a dimer disulfide is there in the structure of cysteine. Cysteine is found in almost most of the proteins. Mainly it gives stability to the three-dimensional structure. And coming next, note down the next one is desmosine and isoleucine. isodesmosine. Okay, desmosine and isodesmosine, these are formed by the oxidation of the cross-linking of the 4 lysine side chains. Okay, desmosine and isodesmosine, which are formed by the cross-linking of the 4 lysine residues of cross-linking of the 4 lysine residues to form desmosine and isodesmosine. These are the constituents component of the connective tissue protein elastin. Next one, phosphoserine, phosphothreonine and phosphotyrosine. Already you know that serine, threonine, both these are the hydroxyl group containing amino acid. So their hydroxyl group is phosphorylated with, means phosphorylated means addition of the phosphoric acid to form phosphoserine, and hydroxyl group is phosphorylated. Hydroxyl group of threonine is phosphorylated to phosphothreonine and phosphotyrosine, and all these are the regulatory proteins. Okay, regulatory proteins means they may be regulate the metabolic, means they may be regulates the other pathways. Next one, selenocysteine. Okay, here in the selenocysteine is here selenium is replaced by the oxygen of the cysteine, and all these are the important modified amino acid. Especially the selenocysteine is present in the enzyme glutathione peroxidase. Glutathione peroxidase protects the RBC membrane from peroxidation. So these are all the modified amino acids which has been formed by the modification of the one of the 20 standard amino acids. Hydroxyproline and hydroxylysine is formed by the modification of hydroxyl, means hydroxylation of proline and lysine, components of the connective tissue protein collagen. Gamma carboxyglutamate, as this amino acid has been formed by the gamma carboxylation of the glutamate. This is quite common in the proteins of the blood clotting factors. Cysteine is formed by the oxidation of the 2 cysteine molecules, which gives stability to the 3-dimensional structure. And desmosine and isodesmosine as the it's a component of the connective tissue protein elastin, and these are formed by the oxidation, cross-linking of the 4 lysine molecules. And all the phosphorylated forms like phosphoserine, phosphothreonine and phosphotyrosine, which has been formed by the phosphorylation of the hydroxyl groups of these amino acids, which are regulatory proteins. Next, selenocysteine, where selenium is the has been oxygen of the cysteine has been replaced by the selenium to form selenocysteine, and this is the component of the glutathione peroxidase. Okay, that's all about the non-standard amino acids or modified amino acids. Then coming to the next one, another type of amino acid is derived amino acids, or else you can call it as non-protein amino acid. Okay, please note down here the another type of amino acids is derived amino acids, or else you can call it as non-standard amino acids, means non-protein amino acids. Okay, please note down this as derived amino acids are nothing but non-protein amino acids. What do you mean by non-protein amino acids? Means we already know that there are 300 different types of 300 means number of amino acids may be found in the cells, but out of the 300 amino acids, only 20 are the constituents of the proteins. The other amino acids are there, they may be available in the cells, but they are not the constituents of the protein. Okay, see here 300 amino acids have been identified in the cells. Out of the 300, only 20 amino acids, standard amino acids, are the constituents of the protein. The rest of the amino acids are they are not the constituent amino acids, but they may be available in the cells and they have variety of functions. So those amino acids which are not the constituents of the amino acids are called non-protein amino acids, or else you can call it as derived amino acids. So here these derived amino acids are found in both plants, and which are found in the proteins are like hydroxyproline, hydroxylysine, gamma carboxyglutamic acid, methylated and acetylated amino acids. Okay, now here first here these derived amino acids are which are few derived amino acids are found in the proteins and few are not found in the proteins. So which are not found in the proteins, you can call it as non-protein amino acids. Okay, see here derived means these are the amino acids which are derived from one of the 20 amino acids. Okay, so here here in the derived also, few may be found in the proteins, few may not found in the proteins, which may not found in the proteins, the B, this one, okay, which may not found in the proteins, you can call it as like non-protein amino acids. So examples of derived amino acids which found in the proteins are, already I mentioned, hydroxyproline, hydroxylysine, gamma carboxyglutamic acid, methylated and acetylated amino acids. This already mentioned under the modified amino acids. Now coming, this one is here, our interest is here to know this, what are the non-protein amino acids. So these non-protein amino acids are found in the cell, but they are not found in the proteins. Examples of the non-protein amino acids are ornithine, citrulline, homocysteine, thyroxine. Okay, and this ornithine, citrulline, homocysteine and thyroxine. And ornithine and citrulline, these two are the components of the intermediates of urea cycle. And the homocysteine as the it is found in the metabolism of the methionine. And the next one, thyroxine. Thyroxine is one of the thyroid hormone. We all know that thyroid hormones has it exhibit function in each and every cell of the body. And along with that, even you can add the homoserine, okay, not homocysteine, and you can add the other examples, homoserine. And the homoserine is involved in the metabolism of threonine, aspartate and methionine. So all these amino acids which are not found in the proteins are non-protein amino acids. Okay, as they are not found in the protein, they may perform variety of functions. Ornithine and citrulline, they are the intermediates of urea cycle. Homocysteine is involved in the methionine metabolism. Thyroxine is one of the thyroid hormone. Homoserine, it is involved in the metabolism of threonine, aspartate, etc. Okay, and some non-alpha amino acids are there. Example is beta alanine and gamma amino butyric acid. These are non-alpha amino acid. Examples are beta alanine and gamma. Gamma stands for gamma amino butyric acid. It's an inhibitor neurotransmitter. Now here, then coming to the importance of these amino acid derivatives. Okay, so already we know that some amino acids are like modified amino acids or derived amino acids are there. So what is their importance means? So GABA, already I mentioned, GABA is derived from the glutamic acid. Okay? From the amino acid glutamic acid, GABA is derived, that is gamma amino butyric acid. And dopamine is derived from the amino acid tyrosine. So these two are the neurotransmitters. Please note down, these are the importance of the derivatives of the amino acid, not the importance of the amino acids directly. The derivatives, the compounds which are derived from the amino acids has the importance, biological significance. So that is the GABA is derived from glutamic acid, and dopamine, okay, that is derived from the amino acid tyrosine. Both are neurotransmitters. And histidine, from the amino acid histidine, histamine is derived, and it is the mediator of allergic reactions. In response to the allergies, histamine is released. What is histamine? Histamine is derived from the amino acid histidine. And thyroxine, it's one of the T4, thyroxine is T4, thyroid hormone, it is derived from the tyrosine. And you know the functions of the which regulates the body temperature, and thyroid hormones exhibit its function in each and every cell of the body. And cycloserine, it is a derivative of serine. It is an anti-tuberculosis drug. Please note down this. And histidine residues, okay, histidine amino acid, these residues which are present in the protein, they may provide buffering activity to the proteins. And ornithine and citrulline, these are the derivatives of arginine, okay, from the amino acid arginine. Ornithine and citrulline are derived, and which are essential for urea synthesis. Okay, so when the question non-standard amino acids, what name the non-standard amino acids and significance means, you have to mention the first names of the non-standard amino acids like GABA, histamine, thyroxine, cycloserine, ornithine, citrulline. These are the non-standard amino acids or non-protein amino acids, which are not the constituents of the protein. And you can write the importance of this one. Okay, these GABA and dopamine are the neurotransmitters. Allergic response, in response to the allergies, histamine is released. Thyroxine is a hormone, thyroid hormone derived from the tyrosine. Histidine, buffering capacity of the protein is due to the presence of histidine, and ornithine and citrulline, they are derived from the arginine involved in the urea synthesis. Okay, then coming to these are the, if you want, note of the list of the derived amino acids or non-protein amino acids. Okay, ornithine, citrulline, homocidine, homocysteine, taurine. You please note down here another non-protein is taurine, which help in the conjugation of the bile acids. And thyroxine, dopa is dihydroxyphenyl alanine and 5-hydroxytryptophan. It is a dopa is a dihydroxyphenyl alanine and this is 5-hydroxytryptophan. All these are the neurotransmitters. So that's it about the derived amino acids or non-protein amino acids. Now I think you can able to differentiate what are standard amino acids, what are modified amino acids, and what are non-protein amino acids. Now coming to the properties of the amino acids. What are the properties which has been exhibited by the amino acids are like, it will be discussed under two headings: physical properties and chemical properties. Physical properties means first solubility, like whether the most of the amino acids are soluble in water, but few amino acids which are hydrophobic, they may be soluble in organic solvents. And amino acids are having their they may be having their particular melting points, and usually amino acids are tasteless. And the next physical property is isomers. Isomers means all the amino acids except glycine, they may possess isomers due to the presence of asymmetric alpha carbon atom. Okay, see here, out of the 20 isomers, alpha amino means amino acids can exhibit isomerism except glycine, because glycine has no asymmetric carbon atom. What do you mean by asymmetric carbon? If 4 valencies of the carbon, if they are attached to 4 different groups, that carbon is said to be asymmetric. So as the glycine, if you remember the structure of the glycine, common features is there may be NH2 group, amino groups, carboxyl group, hydrogen, but in the side chain, even in the glycine, hydrogen is there. So as the 4 valencies has not been attached to 4 different groups in the case of glycine, so glycine has no asymmetric carbon. So as glycine is not having asymmetric carbon, it cannot exhibit the isomeric property. Except glycine, the remaining 19 amino acids they may have asymmetric carbon, so they may exhibit isomeric properties. And the next one is the amino acids. They are the see here, to understand this one isomerism. So this is the alpha carbon. Okay, and alpha carbon means to the carbon where one side there may be amino group is there, the other side carboxyl group is there, that carbon is said to be alpha carbon. And so here like optical activity means amino acids are already mentioned that which are having asymmetric carbon atom exhibit the optical activity, and glycine has no asymmetric carbon, so therefore no optical activity, and all other amino acids are optically active. And these optically active means here these are the D and L isomers. I'll show you the picture. So this is the D and L isomers. So D and L isomers means here this is the alpha carbon in the D form, NH, see here in the D form to the alpha carbon, NH3 is on the left side and carboxyl group is on the right side. Okay, especially its mirror image. Okay, mirror image of D isomer is L isomers. So these two are the isomeric properties of isomers of the amino acid, D form and L form. So these D and L isomers are mirror images of each other. So how could you differentiate D form and L form? Means based on the orientation of the amino groups and carboxyl groups around the alpha carbon. Okay, to the alpha carbon, if NH3 is on the left side and the carboxyl group is on the right side, that is D form, its mirror image is L form. And please remember, under in biological fluids, always this amino acids exist in L form. In biological fluids, this amino acids, they may exhibit in L form. In L form, what may be there? On the left side there is okay, see here to this side NH3 is there on the left side and alpha carbon is there on the right side. So this amino acids exist in L form. Now here optical activity and the mirror images are formed with the already produced with the reference to the alpha carbon atom, that is D and L form. And please note down, L amino acids are occur in nature and all natural amino acids are L isomer only. Okay, that is the optical activity of this one. And the next property, physical property, is amino acids are ampholites. Ampholites means they are amphoteric in nature because amino acids, most of the amino acids are monocarboxylic. So that means they may contain both acidic groups COOH groups and basic groups NH2 groups. So that means because of the presence of acidic means ready to donate the proton, as they contain the basic groups NH2 ready to accept the proton, hence amino acids are regarded as they may be considered as ampholites, or amino acids are considered as amphoteric in nature because they may contain carboxyl group and the carboxyl group and amino groups. Amino groups are ready to accept proton and carboxyl group, acidic group, ready to donate. So as the amino acids are, they may contain both acidic properties and basic properties, so they may be considered as amphoteric in nature. So these are the physical properties: solubility, melting point, taste, isomers, D and L isomers how they have been formed in relation to the spatial arrangement of this NH3 and COO around the alpha carbon. Naturally occurring amino acids are L form, and as they are monocarboxy amino acids are amphoteric in nature. Okay, and here next one is here yeah, if you want you can see this picture. So carboxylic acids are like according to the Brownstein and Lowry. So acids are, what is the property of an acid? They are ready to donate the proton in aqueous solution. So the pK of the carboxylic acid is normally around 2 to 5. So that is the pH at which these acids are 50% ionized. See here, carboxylic acid, what is the property of the acid? Acids are acids are proton donors. Proton donors means in the presence of acids, the hydrogen ion concentration is increased in the solution. Why? Because acids are proton donors. So the pKa is the dissociation constant of the carboxylic acid is normally around 2 to 5. So that is the pH that is the pH at which these acids are 50% ionized. And here see, this is the carboxylic acid. So as it is a being, it exhibits acidic nature, it is ready to so by donating its one, this will become carboxyl. So pH less than means at the acidic pH they may exist like this. Okay, and the next one is amino groups. They may function as bases. So bases mean they are ready to accept the proton. So the dissociation constant, okay, of pK for the amino sugar is usually for amino groups is usually around 9 to 10. So again at the pK of these groups are at that pKa these groups are 50% ionized. So this is the R chain, and these amino groups, as it is ready to accept this one, this will become this. And also pH at below 5 and below 8 they may exhibit like this. Above 9 in the alkaline condition they may exist like this. Okay, so find out that. So try to differentiate this one at pH. Okay, at which pH these ionizable groups, how they are existing. Now here, and the pKa values of the amino acids varies. So this is the list of the pKa values for the some amino acids. For alanine, its pKa at 25 degrees. It's alpha carbon. Okay, the pKa of this alpha carboxyl group is 2.3 and alpha amino group is 9.9. Okay, please note down. For glycine it is 2.4, 9.8. For phenylalanine 1.8. Its pKa value of this alpha carboxyl group, and for this one is 9.1. So like that, please note down the list of these pKa values. Now here, so what is the summary of this one is, so what you have to understand is at low pH. Okay, low pH means what happened? Acidic pH. There is hydrogen ion concentration is high. Okay, low pH means acidic conditions. As the acids are hydrogen donors, at low pH there is increase of hydrogen ion concentration. So therefore both amines and carboxyl groups acids are protonated. at high pH, what happened? Protein concentration is low. High pH means under alkaline conditions, the protein as the bases are hydrogen proton acceptors, there is decreased concentration of hydrogens. So therefore, both amines and carboxyl groups of the amino acids are deprotonated. At neutral pH, amino acids are protonated and carboxylates are deprotonated. That is the summary of this one. Okay, again I repeat, at low pH, under acidic conditions, there is increased concentration of the hydrogens. So therefore, both amines and carboxyl groups of the amino acids are protonated. Okay, like this. And here at high pH, okay, that is high pH means alkaline conditions where hydrogen ion concentration is low, there both amino and carboxyl groups of the amino acids are deprotonated. At neutral pH, amines are protonated and carboxylates are deprotonated. Means at neutral pH there is no charge. Okay, and there is at neutral pH there is no charge. Okay, and the next one is zwitterion. So zwitterions means here, as the most of the amino acids are diamino dicarboxylic acids, so that means where it has equal number of positive and zwitterions means here at the neutral pH. At neutral pH, already we have discussed that these amino acids exist as carboxyl group and like this. So it may carry negative charge and positive charge. So at neutral pH, ultimately there is these amino acids, they may exist as zwitterion. So zwitterion means the pH at which the protein exists as zwitterion. So zwitterion means where it has equal number of positive and negative charges. So there is no net charge at this pH. It means if it exists as zwitterion means that indicates that it has equal number of positive and negative. So that restrict the movement of the amino acids under electric field. At that pH, the amino acids are least soluble and gives the maximum precipitation. Okay, I repeat, zwitterions. Zwitterions means ions bearing the two charges were named as zwitterion. Zwitterion means at pH, at physiological pH 7.4, proteins exist as monoamino monocarboxylic. Okay, like monoamino monocarboxylic means there may be equal number of the positive charge, and because of the amino groups it attains positive charge. Because of the carboxyl group it attains negative charge. So at physiological pH, the amino acids have equal number of positive and negative. So there it exists as zwitterion. And the property of the zwitterions is where the amino acids are least soluble and gives maximum precipitation. Okay, so now here and you all know that amino acids because they are so here the next one is we have to understand. So what we have to understand is here, these amino acids, due to the presence of ionizable groups, they may behave as acid base and zwitterion, and they may attain the buffering activity. Again I repeat here, because the amino acids, if you see the structure of any amino acid, they may be having at least two ionizable groups. Two may be there. One is amino group and is carboxyl group. Because of the presence of ionizable groups, amino acids behave as acids and bases, and they may exist as zwitterion, or they may be having the buffering capacity. So acid, so can you explain, like how the amino acids behave as an acid, how they behave as an base means here all so as the because of the presence of ionizable groups, COH groups and amino groups. For example, you take acidic is because of the presence of carboxyl group, and basic nature of the amino acid is due to the presence of amino groups. So because of the carboxyl group, what happened? Carboxyl group of amino acid can donate. Carboxyl group means acid, carboxylic acid being an acid ready to donate its hydrogen. So it behaves as an acid. So it is forming a negatively charged. For example, you take this ammonia. these amino groups. So these amino groups are being a base can ready to accept the proton and behave as base. So it forming a positively charged. Okay, see here, this is the basic structure of an amino acid. No, see here it may be having all our amino acids are having carboxyl group and amino groups. Carboxyl groups are carboxyl group of amino acids are can donate the amino acid. So it behave as an acid. It gives the negative charge to the amino acid. Now here another one, amino groups. Amino groups can accept the proton and it behave as a base and it is forming positive charge to the cation. So amino acids in aqueous solutions are ionized and acts as acids and bases. Amino acids in aqueous solutions, they are ionized and acts as acids and bases. And what about the pH 7.4? What about the pH 7.4 means at physiological pH, carboxyl groups exist as carboxylic, okay, COH groups exist as COO minus. Amino groups exist as NH3 plus. So that means where at the 7.4 it exists as the Zwitter ion. At physiological pH 7.4, COOH groups exist as COO minus, ionized to COO minus, and amino groups are protonated to and they may be present, they may exist as NH3. So at 7.4 they may exist as Zwitter ion where it has equal number of positive and negative. And at pH 1, so what and Zwitter ion means dipolar ion. Dipolar ions means we know that. So amino acids are monoamino, monocarboxylic. In the solutions, they may be, most of the amino acids are monoamino, monocarboxylic. So in the solutions what happen, they may be exist as Zwitter ion. Zwitter ion means where they have the equal number of the positive and equal number of the positive and negative charges are there. Okay, that's it about this properties. And if you want the more, see here, look at this picture. So here this is the neutral pH, and the neutral pH and high pH and low pH. You just look at this slide. At neutral pH, what happened? There is equal number of the positive and negative charge. And the low pH means under acidic pH, what will happen? See, this carboxyl group is, see here it is converted, so it exists as it is ionized to COOH. And at the high pH, what will happen? Alkaline conditions, these NH3 groups are which is protonated to NH2. Okay, like that. So this amino acids at neutral pH, it exists like this. Under acidic conditions, amino acid behave as a base. Okay, under alkaline conditions, amino acid behave as an acid. Okay, see here, that's what you have to understand at the end. So this at the neutral pH, they exist as Zwitter ion. Okay, so when that amino acid, when you put it in the acidic solution, they may exist as a base. Okay, they may be served as a base, they may behave as a base. Now here, when the same amino acid, if you put it in the alkaline conditions, so they may serve as the acid, means they may they may behave as a base. So now here, like if you want like how the amino acids under acidic conditions and under alkaline conditions, how it exists as cation and anion. Okay, for more again to understand, to explain you clearly, so look at this picture. This is an amino acid, and here high pH under the alkaline conditions, what will happen? It exists as anion. And here, okay, and amino acid under acidic conditions, it exists as cation. And Zwitter ion means where it has equal number of positive and negative charge. Now here again the same picture. So Zwitter ion and conditions under acidic conditions and alkaline conditions, how it exists as cation and anion. Now here then the definition of the isoelectric pH. Isoelectric pH means it is like it is represented by the Pi. So isoelectric pH means it is defined as the pH at which the protein exists as Zwitter ion or dipolar ion. So Zwitter ion means you know that where it has equal number of the positive and negative charge. So ultimately there is no net charge at the isoelectric pH. Isoelectric pH means each protein is having the its own isoelectric pH, that is represented by Pi. Isoelectric pH means at that pH proteins has no net charge, where it exists as Zwitter ion, where it has equal number of the positive and negative charge. And the property of the proteins at their isoelectric pH is they are least soluble and gives maximum precipitation, and there is no movement of this proteins under electric field at their isoelectric pH. For example, you take casein. Casein is a phosphoprotein, milk protein. Its isoelectric pH is 4.6. like that. And now here, so already you know that Zwitter ion. So Zwitter ion means here where it has the equal number of the like positive cations and anions. And amino, for example, the isoelectric pH of the leucine is 6, and which can already you know where it at pH 6 leucine has equal number of the positive and negative charges. Now here ampholites means already you know that proteins are, because as they behave as an acid and bases, because of the presence of carboxyl group and amino groups, they amino acids, they may exhibit their amphoteric in nature. But here the charge on the amino acids, that depends upon the pH of the medium. Okay, please note down the charge which carry on the amino acids, that depends upon the pH of the medium. At isoelectric pH only they may exhibit they may behave as exist as the Zwitter ion. Under low pH it exists as cation, under high pH it exists as anion. Okay, see here the charge on the amino acid in turn depends upon the pH of the medium. Okay, at their isoelectric pH exists as Zwitter ion. Under high pH alkaline conditions, basic conditions, it may exist as anion carries. Okay, and at low pH acidic conditions it may exist as cation. Okay, now here this is the definition of the isoelectric pH, and the solubility and buffering capacity will be minimum at their isoelectric pH. General isoelectric pH is represented by this Pi, and again I repeat, in acidic solutions they are cationic in the form, and in alkaline solutions they exist as anions. You have to please remember this, keep this point in mind. Under acidic conditions behave as cations, under alkaline conditions behave as anions, and at their isoelectric pH there is no net charge, no precipitation, and gives least precipitation, no movement under electric, no mobility under the electric field. So then effective buffer means few amino acids, they have the buffering capacity. So buffering capacity, the pK value of the mainly buffering capacity of the protein, especially hemoglobin, it acts as a buffer. Its buffering activity of the hemoglobin is mainly because of the presence of histidine. One of the amino acid, basic amino acid histidine, is responsible for buffering activity of the protein. Example, hemoglobin, it acts as a buffer. How hemoglobin acts as a buffer means as it is having more number of histidine of the hemoglobin is responsible for buffering. So the pK value of the imidazoles are the functional groups of the histidine is 6.1. So and therefore effective as a buffer at the physiological pH 7.4. Please note down the buffering capacity of the plasma protein is hemoglobin is mainly because of the presence of histidine residues. And what is this pK value? pK value, and already I mentioned that pK is the, as we have already learnt that amino acids have at least 2 ionizable groups. So each ionizable groups, okay, at least 2 ionizable groups, that is carboxyl group and amino group. So each ionizing group has a dissociation constant, that is Ka. Please note down amino acids. We know that most of the amino acids are monocarboxylic. So that means at least 2 ionizable groups. So each ionizing group has a dissociation constant, that is Ka. So acid strength of weak acid may be expressed in terms of their acid dissociation constant, that is pKa. Okay, please note down the acid strength of a weak acid may be expressed in terms of their dissociation constant, that is pKa. pKa is the acid dissociation constant. Now here maximum buffering capacity occur where pH is equal to pKa. I hope you are understanding. Maximum buffering capacity occur where pH is equal to pKa. Okay, so that means pKa is the acid strength of the weak acid, and that generally we may be expressed in terms of the pKa. Okay, and now finally pKa is, what is pKa? pKa is it which expresses the strength of the weak acid, or else it may be the measure of the tendency of a group. Okay, it may be the measure of the tendency of a group to give a proton. Again I repeat, pKa is the dissociation constant and it expresses the strength of the weak acid, or it measures the tendency of a group or to give a proton. So finally what you have to understand is, small pKa value is the stronger the acid, larger the pKa value, weaker the acid. Okay, so the capacity of the strength of the acid can be assessed by its pKa value. Please note down, the low pKa value, smaller the pKa value, stronger the acid, larger the pKa value, weak acid. Okay, that's all about the chemical property like this buffering, as because these are the properties of the amino acids, as because of the presence of ionizable groups at the end. What you have to remember is, as because of the presence of ionizable groups, that is COOH group and amino groups, amino acids, they may behave as acids, bases, they may exist as zwitterion and zwitterions, and then they may act as they may attain buffering activity because of the presence of ionizable groups. Okay, that's all about this one, and this titration, this part is not required, and in the next coming class let me continue with the chemical reactions of the amino acids. Okay, chemical reactions. So till now we have discussed the physical properties various because of the presence of ionizable groups. Then in the next coming class we'll explain you, we'll discuss the chemical reactions of the amino acids. So the chemical reactions may be because of the presence of, again, due to few chemical reactions due to the presence of carboxyl group, few may be due to the presence of amino groups, or few chemical reactions due to the presence of side chain. So what are all these reactions? Let us discuss in the next theory class. It will be okay, the class will be continued in the next class. Thank you.